seeing on the molecular biology of breast cancer. We all know who Angelina Jolie is. Her contribution to the community of oncology is very big. Soon after her diagnosis of BRCA mutation, she underwent risk-reducing mastectomy. That is called the Angelina effect, after which the patients who got diagnosed with this BRCA mutation underwent this risk-reducing surgeries. There was a great increase in the number of surgeries after her diagnosis and her risk-reducing mastectomy. This is called the Angelina effect. So this got published in the Times magazine when in 2013 she got her surgery done, after which there is great increase in the number of surgeries and patients getting diagnosed with this condition. And subsequently she announced that her chances of getting this breast cancer have reduced from 87% to 5% because of this risk-reducing mastectomy. So let's go into the topic per se. So germline mutations in breast cancer. There are around 10% of breast cancers which are due to germline mutations. The breast cancer susceptibility genes can be classified as high penetrance genes, the moderate penetrance genes and the low penetrance genes. This is based on the risk that they contribute to in the breast cancer. So when the risk is around more than 6, it will be high penetrance genes. The risk between 2 to 4 will be called as moderate penetrance genes, and if the risk is less than 2, then it will be called as low penetrance gene. There are 5 high penetrance genes: the BRCA, the TP53, the P10, CDH1 and your STK11. Let's go into the details. The BRCA 1 and 2 are responsible for your hereditary breast and ovarian cancer. They are present in the chromosome 17 and the chromosome 13. The TP53 is responsible for your Li-Fraumeni syndrome, which is composed of your breast cancer, your soft tissue sarcomas, your CNS tumors, adenocortical carcinoma and leukemia. And it is present in the chromosome 17 again. The P10, which is responsible for your Cowden syndrome, consists of your breast cancer and mainly your CNS tumors present in your chromosome 10. The CDH is responsible for your familial gastric cancers, wherein the gastric cancer is of diffuse type, diffuse Linitis plastica type, and also they have lobular breast cancers, which is present in again chromosome 16. STK11, which is responsible for the Peutz-Jeghers syndrome, wherein there are melanotic macules along with your GI hematomas and your cancers present in your chromosome 19. So these are your chromosomes and your genes which are responsible for high penetrance breast cancers. Let's go into the details of BRCA1 and 2. The BRCA1 is present in chromosome 17. The BRCA2 is present in chromosome 13. They are responsible for your hereditary breast and ovarian cancer. These genes, the BRCA1 and 2, are the tumor suppressor genes. They are responsible for DNA repair, mainly your double-stranded DNA repairs. They do it through homologous recombination and not through the other pathway, which is called the non-homologous end joining. These two are the mechanisms of double-stranded DNA repairs, and this BRCA1 and 2 takes the pathway of homologous recombination. This happens with the help of other proteins like your PCNA, RAD50 and RAD51. So the DNA repair is mainly for your double-stranded DNA breaks or your single-stranded DNA breaks. When there is a double-stranded DNA break, it can be through your homologous recombination or your non-homologous end joining. What is homologous recombination? As the name suggests, there is homology between your broken DNA and another neighboring DNA. What happens after a DNA damage? Once a DNA is damaged, there can be a double-stranded DNA break. This double-stranded DNA break gets corrected by two mechanisms. Those are the homologous recombination and the non-homologous end joining. What is non-homologous end joining? It is just a joining of the cut ends by which the cut ends get cut further and then gets joined without help of any other DNA. The problem here is we can lose few base pairs and subsequently there can be frame shift or loss of function mutations. So this is a error prone mechanism. But what happens in your homologous recombination. There is help from a neighboring homologous DNA, which helps this broken DNA to get its base pairs and subsequently corrects its brokenness. That is how homologous recombination happens. And the proteins which help in this homologous recombination are those which are BRCA1 and 2 along with the other proteins. This is how BRCA1 and 2 help in homologous recombination. So, any BRCA1 and 2 mutation will be called as homologous recombination deficiency or the HRD. So, let's go into the HRD or the homologous recombination deficiencies. This BRCA1 and 2 mutations are inherited as autosomal dominant forms. There can be n number of mutations, more than 1000 mutations, which can be present in your BRCA1 and 2 genes. And because of this mutation, the most common problem is your truncated protein products, which prevents the mechanism or the function of your BRCA1 and 2 proteins. And also there can be mutation that interfere with this protein function. Either the protein is absent or it is functionless. These two are the mechanisms by which they lose their function. But then not all members of the family who carry this BRCA mutation develop the cancer at the same time and develop the same cancers. Why is this variation in the risk? The risk variation is because of the gene-gene interactions, the gene to the environmental interaction, and also the difference in the specific mutations. Also, there are genetic modifiers, that is the single strand or the single nucleotide polymorphisms. These are other mechanisms by which there is variation in this risk even though the patients carry this BRCA mutation. The variation between the family members is because of these factors. So, what are the features of a BRCA1 related breast cancer? They are usually young and they have aggressive features in their breast cancer, like your high grade, high Ki67. What is Ki67? Ki67 is nothing but your proliferation marker. A high Ki67 denotes that the proliferation index is very high. The other proliferation index is your MIB. The most commonly used is Ki67. Ki denotes kill, wherein that is the area where it was discovered. It's a IHC marker for proliferation. And the other feature is absence of hormones and the HER2 receptors. These are the features of a BRCA1 related breast cancer. Also, they have a basal-like expression profile, means they express basal-like proteins like your cytokeratin 5/6, cytokeratin 14/17, EGFR and your p-cadherin. These are the set of characteristics a patient who has a BRCA1 related breast cancer expresses. Also, a patient who has a BRCA1 mutation or a BRCA2 mutation are sensitive to platinums and the PARP inhibitors. Why are they so sensitive? Is because of the mechanism of the platinums. We know the platinum, the most commonly used platinum is your cisplatin and the other is your carboplatin. The cisplatin, as we see here, is this is the platinum which has two chloride groups and the two ammonia groups. The chlorides get attached to the DNA and cause interstrand and your intrastrand crosslinks, by which they cause double-stranded DNA breaks. As we saw, this double-stranded DNA break has to be repaired by either your homologous recombination or your non-homologous end joining. This non-homologous end joining contributes very less to your double-stranded breaks. So, it is a less commonly used pathway. The most commonly used pathway for repair is your homologous recombination. So, any patient who has a BRCA1 mutation, 1 or 2 mutation, will be deficient in this homologous recombination, and henceforth there is ultimate death of the cells, causing increased sensitivity to the platinums. This is how they are more sensitive to platinums. And what is the other drug which are more active in this BRCA mutated patient is the PARP inhibitors. What is PARP inhibitor? As we know, a DNA which has continuous replication, there are single strand breaks. This single strand breaks, if not repaired, progresses to double strand breaks. How is the single strand break getting repaired? Is through base excision, nucleotide excision and other kinds of repairs. One of the most important repair is your base excision repair. The bases get repaired as and when they get damaged. One of the main factors for this base excision repair is your PARP protein. So once there is a break in the bases, the PAR proteins get along with it and causes repair of these cells. When you inhibit this PAR, there is no repair of this single strand break. Ultimately, this single strand break progresses to your double strand break. As we have already seen, any patient who has your double strand break along with your HRD, that is homologous recombination deficiency, because of loss of BRCA1 and 2 proteins, are progressing to cell death. This is how a patient with BRCA mutation is also sensitive to PARP inhibitors. So they are sensitive to both platinums and PARP inhibitors. So what are the PARP inhibitors? PARP inhibitors are the drugs named as olaparib, niraparib, rucaparib, talazo and veliparib. The first drug which was approved was olaparib, based on a trial called Olympiad trial, and which gave an improvement of around 2 to 2.5 months in their progression-free survival in metastatic breast cancers who are BRCA positive. Other drugs are under development and yet to be approved by your FDA. The other basal-like non-BRCA related breast cancers are also sensitive to this kind of PARP inhibitors. So let's go on to the risk reducing strategies. So far we have seen what are the mechanisms by which the DNA gets repaired. How does BRCA help in this DNA repair? What happens if this BRCA gets mutated? And what are the features of a patient who has a BRCA mutated breast cancer? And now we will go on to treat patients who are BRCA mutated. There can be patients who are having this BRCA mutations along with their siblings who are also mutated. So once we have diagnosed a patient with the BRCA mutation, we are supposed to screen the family for such a mutation. And once you have screened the family and you have picked up other members who are having this BRCA mutation, we are supposed to offer them risk reducing strategies. That is, risk reducing their development of breast cancer or an ovarian cancer. So what are these risk reducing strategies? The strategies can be either invasive or non-invasive. Whom are you going to perform all this risk reducing strategies? It's mainly for the patients who are at high risk of developing this breast cancer. Who are these high risk patients? Who have a very strong family history of breast cancer, who are already mutation positive, or has more than 20% lifetime risk of developing a breast cancer. What is this lifetime risk? We'll be discussing in the next slide. Let's go on to find out what are the risk factors of this breast cancer. And the risk factors of breast cancer can be divided into 3 columns based on the relative risk. A relative risk of less than 2, 2 to 4 and more than 4. This carries the most important and the most high risk of developing a breast cancer. And the 4 main causes or the risk factors are mutation in BRCA1 and 2, your LCAS or your lobular carcinoma in situ, atypical hyperplasia, and any radiation exposure for a patient less than 30 years of age, mainly your Hodgkin's lymphomas who get mantle cell radiation at their 25 or 30 years of age. What are the other risk factors? Let's see that also side by side. The risk factors which are having a relative risk less than 2 are those mainly which causes increased hormonal circulation in the body. That is, increase in estrogen in the level and the estrogen exposure for a patient, like early menarche, late menopause, patients who are nulliparous, or intake of estrogen and progesterone pills and your hormone replacement therapies. These are the conditions wherein there is increased time and exposure for a patient for their estrogen levels. Also, a patient who has postmenopausal obesity tend to have more risk than your premenopausal obesities. Premenopausal obesities have problems with their hormonal surge because of obesity they have disturbances in their hormonal production. That is why they are at a decreased risk. Whereas in postmenopausal obesity, these patients have peripheral production of hormone, that is estrogen, and therefore they are at an increased risk of this breast cancer. And others like your proliferative breast diseases also contribute to a risk for your breast cancer. Now let's discuss mainly your high risk factors, that is your mutations, your radiation, your atypical hyperplasia and LCIS. So for such patients, you are going to offer your risk reducing strategies. Along with your BRCA mutated patients, you are also going to offer this risk reducing strategies for other patients who have high risk of developing breast cancer, like other 3 factors which we have seen now. So what is the risk model or the risk lifetime risk which we have dealt now? More than 20% lifetime risk has come from a model which is either a Gale model or a Claus model or the other newer models. So let's see about the risk models. There are models which are useful for finding out the lifetime risk of breast cancer. The most common and the most ancient model is your Gale model. The other subsequent models which came up next are the Claus model, the Tyrer-Cuzick and the BRCA-Pro. The most comprehensive model is your Tyrer-Cuzick model. The most commonly used and commonly asked in exams is your Gale model. What is this Gale model and where does it come from? It comes from your breast cancer reduction demonstration project study. The Claus model comes from your cancer and steroid hormone study. What does this model include? This Gale model includes your first degree relatives with breast cancer, whereas your Claus model includes both first degree and second degree relatives with your breast cancer. Also, Claus model includes your age of onset of this breast cancer in the relatives. Apart from this, Claus model includes only the age of onset of risk in the person whom you are calculating the risk. But in Gale's model, there are other characteristics like age at menarche, age at first live birth, number of breast biopsies, atypical biopsies in the past, and also the race. Gale model is more important because it includes risk factors other than the family history. Though in family history, it includes only the first degree relatives. The Claus model includes also family history of ovarian cancer. So these are the characteristics of a risk model through which we calculate the lifetime risk of developing a breast cancer in a patient. And if the lifetime risk is more than 20%, we can offer them risk reducing strategies. So what is the risk of development of a breast cancer or an ovarian cancer in a patient with BRCA mutation? A BRCA1 mutation carries the higher risk than your BRCA2 mutation, and breast cancer at a higher risk than your ovarian cancer. So breast cancer at around 60% in one and around 50% in BRCA2 mutation. There is also a patient who has a BRCA mutation and who has already developed a breast cancer on either of the right or left side of the breast, has a higher risk of developing a contralateral breast cancer to the tune of around 83% in BRCA1 and around 62% or 60% in your BRCA2. And the ovarian cancer risks are very high with BRCA1 mutation compared to your BRCA2 mutations. Apart from your BRCA mutation, other high-risk penetrant mutations also benefit from your risk-reducing strategies. So, all other high-risk mutations also benefit from your risk-reducing strategies. We will go on to the strategies of this risk reduction. We have seen that it can be either non-invasive or invasive. So, when do you do this non-invasive risk reduction? Is for patients who do not want a surgery or who wants to delay the surgery for a certain period of time. So, these are the patients who will be offered your non-invasive high-risk surveillance and your chemo prevention. What are the non-invasive strategies? Those are the high-risk surveillance and chemo prevention. High-risk surveillance is not your breast self-examination, annual mammogram or the clinical breast examination. These three are not the modalities of high-risk surveillance for reducing the risk of your breast cancer. So, high-risk surveillance mainly includes the MRI along with mammogram. Mammogram in separation will not be as a surveillance modality, but along with the MRI, mammogram is more important, and MRI alone is also not a screening modality for your high-risk surveillance. So, MRI increases the sensitivity for detecting a breast cancer, and mammogram is more specific for detecting a breast cancer. This is how both of these modalities are combined for us to find out whether the patient can develop a breast cancer or has developed a breast cancer. So, how are this MRI and mammogram performed? They are performed annually. So, how are they performed? They are performed at a 6-monthly interval. So, when you do an MRI, the next 6 months you do a mammogram. In the next 6 months, you do a MRI again. This is how they are spaced, and we get your interval progressions or interval appearances of your breast cancers. And this is one of the major modalities of your non-invasive surveillance or a non-invasive risk reducing strategies. So, when do you start such surveillance? In a patient with BRCA1 and 2 mutation, you start at the age of 30. You never start below the age of 25 and you do yearly checkups as we have seen now, like MRI and mammogram at 6 monthly interval alternating. But in a patient who has undergone radiation, for example, a patient with Hodgkin's lymphoma who has undergone a mantle cell radiation, the age should be at least 25 to start with, or 8 years after the period of radiation. And if they have received at least 20 grays of your radiation, they are the patients whom we are supposed to do this surveillance. The benefit of this high risk surveillance in LCIS patient is controversial. So again, let me remind you, the high risk or the relative risk more than 4 for a patient to develop breast cancer where the BRCA mutation, the LCIS, patients who have undergone radiation, and also atypical hyperplasia. So these are the patients whom we have seen how and when to start your high risk surveillance. That is one of the non-invasive modalities for risk reducing strategies. So next non-invasive modality is your chemoprevention. Chemoprevention, as the name denotes, it's not a chemotherapy. It is your endocrinal therapy wherein tamoxifen can be used for a premenopausal woman, and postmenopausal either your SERMs, the tamoxifen, raloxifene, or your exemestane. The problem of such drugs are the thrombotic complication and a very small absolute benefit which is obtained through this drugs. But a patient who really wants to delay their surgeries can be taking such drugs for delaying their onset of breast cancer. So the major non-invasive methods are your chemoprevention and your high-risk surveillance. Let's go on to the invasive method. So the invasive method of risk-reducing strategies. So what are the invasive method? Those are the surgeries as we know. The risk-reducing mastectomy and the risk-reducing salpingo-oophorectomy. It includes both your fallopian tubes and the ovaries, salpingo-oophorectomy. Here we are seeing the invasive methods to prevent breast cancer. But the risk-reducing salpingo-oophorectomy is also helpful for reducing your ovarian cancer. So it's an additional benefit which is obtained through this RRSO. So what are the methods by which we do your risk-reducing mastectomy? Risk-reducing mastectomy. It can be a total mastectomy or a simple mastectomy, a skin sparing mastectomy, or a nipple sparing mastectomy. All these modalities are equal in terms of their survival benefit. Henceforth, a nipple sparing mastectomy is more cosmetically appealing for us, and this is more commonly practiced nowadays. The risk reduction with intact ovaries is around 90%, but when a patient has undergone a prior oophorectomy, the risk reduction increases to around 99%. So oophorectomy, by reduction in the hormonal production, helps in reducing the risk for a development of a hormone positive breast cancer. As we have seen already, the risk reducing salpingo-oophorectomy also helps in additional reduction of your ovarian cancer. But why do you remove your fallopian tubes along with your ovaries, and how does it help in ovarian cancer? The current theory of development or the origin of ovarian cancer is STIC or the stick, that is the serous tubal in situ carcinomas. Means that the fimbrial end of the tubes are probable origin of your ovarian cancer. So the current modalities or the current development and the future is to find out if we can remove only the tubes and not the ovaries, so as to find if they are helping in prevention of your ovarian cancer. That is the reason for removing both your tubes and the ovary as of now. So these are the methods of risk reduction. So far we have seen a non-invasive method which includes your high risk surveillance, the surveillance through your MRI and mammogram, and also the other non-invasive method, the chemoprevention, which is through your drugs, tamoxifen and your exemestane. And the invasive methods which we have seen are the risk reducing mastectomy and the risk reducing salpingo-oophorectomy. So these are the modalities by which we prevent the development of breast cancer in a patient who has been positive for your BRCA mutations or other high risk factors which put them at a higher risk of developing their breast cancer. Now let's go into the other molecular factors or the molecular aspects of the breast cancer. What are the clinical subtypes of breast cancer? The current clinical subtypes which we are following are the hormone positive breast cancer, the HER2 positive breast cancer, the triple positive or the hormone and the HER2 positives, and the triple negative which are negative for your ER, PR and your HER2. So what is transcriptional profiling of your breast cancer? Having said what are the clinical types, let's go on in detail about the transcriptional profiling of your breast cancer. We all know that there is a DNA. From DNA comes your mRNA. The mRNA gets translated to your proteins. So this is the normal pathway by which a DNA forms your proteins. By finding out how are this mRNAs or the transcripts get expressed, by which we call it as gene expression profiling, we can diagnose a breast cancer, we can find the prognosis of the breast cancer, and we can also decide about what treatments can be individualized for such patients. Let's go into details of these same. So how do you do gene expression profiling? Let's go into the depth of this. See this picture once and then I'll tell in detail about how a gene expression profiling is done. Let's take a normal cell and a cancerous cell. From these cells, you get a DNA first, you extract the DNA from this cell, then you make the mRNAs which are there in the cells and you extract the mRNAs. So once the mRNAs are isolated, then you do in lab, that is in your lab culture, you do your cDNA or the complementary DNA, that is through a reverse transcription. So you do a reverse transcription of the mRNA from the normal cell and the cancerous cell. Once this cDNA or the complementary DNA are formed, then you put it into a chip, that is your microarray, which is already having the complementary strands for this complementary DNA. So if this DNA has a complementary strand in your tray, then it goes and combines. If it does not have, it will not combine. The same way, from a cancerous cell, if this cDNA has a complementary strand in your tray or your microarray, then it combines. If both of them will have a complementary strand in your microarray, then both of them will combine with that. That is how it is being made, or the microarray is getting expressed. So sometimes if it never combines, then there is gray color that is not present in the cell. So the corresponding cDNA is not present in either the normal cell or your cancerous cell. If a DNA which is present in both normal cell and your cancerous cell is being expressed and that gets combined with the complementary DNA in your array, then it shows your yellow color. It means it is present in the gene, or the DNA is present in both your normal cell and your cancerous cell. According to the color of the normal cell and the cancerous cell here, the normal cell expresses DNAs, and if it gets combined with the complementary DNA in the array, then it will be green in color, and when it is from the cancerous cell, it will be red in color. So this is how you do a gene expression profiling. Now you know what are the genes which are expressed in the tumor, what are that which are expressed only in the normal cell, and what are the genes which are present in both the normal cell and the cancerous cell. This is how you do a gene expression profiling. So now you know the profile of genes which are present in your tumor samples. So that is how you do a gene expression profiling, and this is through a technique called microarray. So through this microarray technique, what you have done is gene expression profiling. How is it useful? It is useful in predicting the tumor behavior. This molecular signatures, that is a tumor and its specific genes, that is the genes which are expressed only by these tumors, which is not present in the normal cell, is helpful in improving the diagnosis. It is predicting for your recurrence, and also it helps in selection of therapies for individual patients. You can do it through either a microarray or a next generation sequencing, that is your RNA sequencing, or a multiplex PCR. These are the techniques by which you can get a gene expression profiling done. So, how does this gene expression profiling being presented to you, and what happens in your breast cancer tumor which has undergone gene expression profiling? Once a gene expression profiling of a tumor is done, this is how it looks to us. So, this is your gene expression profiling of your breast cancerous tumor or a breast cancer cell. So, here the blue denotes your luminal A subtype. I will discuss in detail of each of this. The blue, this column of blue denotes your luminal A subtype. This orange denotes your luminal B subtype. The normal like is seen in your green color, and those which are HER2 positives are seen in your pink color, and the red ones are your basal like subtypes. So, this set of genes are expressed in luminal subtypes. This set of genes will be expressed in your luminal B subtype, this for your normal, and this set of genes for your HER2 positives, and this set of genes for your basal like subtypes. Let's go into detail how it is being done. Let's understand the basics of what this gene expression profiling presentation is being made. This is through a method called as hierarchical clustering. Let's go into details of genetics and how it is being made, so as to understand how it is being presented to us in your microarray DNA clustering and your DNA sampling kind of thing. So, here in hierarchical clustering, it means it goes through a hierarchy. Let's see here. So, each sample from a patient was studied retrospectively initially, and then they have calculated the amount of genes expressed in each of these blood tumor samples, and they have profiled it for each of the tumor samples, and then combined the common ones into a single group and the other common ones in the next group, and that is how the hierarchy is set and a group is formed, ultimately leading on to your molecular subtypes of your breast cancer. So, the initial groups might be 5 groups. Among this, the 3, 4 and 5 might be looking the same. They will be called as single cluster, and the 1 and 2 might have looked the same. They will be called as the second cluster or the cluster A. That is how the similarities are being classified and clustered, and hierarchically they are being arranged so as to form your ultimate molecular subtypes of your breast cancer. So, for example, take this picture. In this picture, this is ABCDEFGH, where FGH is a group and ABCDE is a group. Among this ABCDE, A was a separate group, BCDE was a separate group. The BCDE again had a DE and your BC, and ultimately leading on to all these samples. The hierarchy was built from down. We know that all the samples are there, and based on the similarities, we built up the hierarchies and ultimately classified and clustered them. That is how, with the presence of genes and the expression profiles, that you combine all these samples, and then you progressively arrange them to form this hierarchical clustering, and then classified them into luminal A, the luminal B, the HER2 like and your basal like, which are important for you clinically. So, these are the molecular subtypes of breast cancer. So, let's go into each of these subtypes. So, what are the luminal subtypes? Luminal subtypes contribute to 70% of the breast cancer subtypes. They express genes that are normally expressed in your luminal epithelium. That is why they are called as luminal subtypes. What are those genes? They express genes called as ESR1, the CK818, GATA, FOX and so on. So, these are the genes normally expressed in your luminal epithelium, and a tumor which expresses all these genes will be called as a luminal subtype. So, this is again divided into your luminal A and your luminal B. The luminal A has high ER related genes, low HER2 related genes, low proliferation genes. Means what? Let's go here. So, the luminal A here will have more of your ER genes. So, those genes which are responsible or related to your hormone receptor, that is your estrogen receptor, the genes related to that will be expressed more in higher number in your luminal subtype A. Those genes which are related to your HER2 will be expressed very low in your luminal A. So that is how it is. So high expression of ER related genes, low expression of the HER2 related genes, and very low expression of your proliferation associated genes. There are also genes which are associated or which are responsible for the proliferation characteristic of the tumor cell, and that is how they are being formed, and there is a low expression of such proliferation genes in your luminal A subtype, and it has a very better prognosis compared to your luminal B. The same way, when you come to your luminal B subtype, ER related genes are very low, but they are still expressed. The HER2 genes are variable. It can be high, it can be low, or any of this. But the proliferation genes are very high. So it is very high proliferation genes with low expression of your ER related genes and your variable expression of your HER2 related genes. Just not to confuse, let me show the picture again. It means that the luminal A, this subset of patients have, so this luminal A has all your genes which are which express much of your ER related ones and express low of your HER2 related ones. Let's see here. So the HER2 related ones, when they are present in red color, they are less expressed, and when they are present in green color, they are more expressed. That is how they are being arranged here. So let's go into the next subtype, that is the luminal B, which we have seen now. This compared to luminal A has a worse prognosis. So clinically, this is helpful as a luminal A and B in mainly node negative patients. That is, when they are axillary node negative, a tumor which can be either a T2 or T3, which is usually node negative, your luminal subtypes are useful clinically for making decisions. Ka67, 15% is taken cutoff for differentiating between your luminal A and luminal B. So a patient who has T2N0 M0 who has Ka67 of 30% will be called as luminal B. On the other hand, who has a 8% Ka67 will be called as luminal A, who is supposed to have ER positive and HER2 negative. So this is how they are being utilized clinically. They are more important for the prognostic purposes, and also they predict the response to the endocrine therapy. So this completes the luminal molecular subtype of your breast cancer. Coming to the next subtype, that is the HER2 subtype. So HER2 subtype also has genes which are expressed. So it has very low expression of your ER related genes, as we know, and it, as the name suggests, should have overexpression of your HER2 related genes and overexpression of your proliferation associated genes. It is present in around 10 to 15% of breast cancer, but we have seen that clinically we call a patient as HER2 positive when they are IHC3 plus or if their FISH is positive, but not all these patients fall into the HER2 enriched subtype, and the vice versa is also true. So there can be a patient who is IHC3 positive or who is FISH positive, and their gene expression profile might not be as HER2 enriched. So this is not very direct of your clinical correlation. On the other hand, a patient who is IHC negative and or your FISH negative can have the gene expression profiling which are indicative of HER2 enriched subtype. So this is very clear here. And those who are not HER2 enriched, who are still IHC3 positives, can be the other subtype called luminal B. We have seen in that condition luminal B that the HER2 cluster can be variable. So if a patient is HER2 negative, clinically that means either IHC or your FISH negative, but then they are still not in HER2, they are positive and they are not in HER2 enriched subtype, will fall into your luminal B subtype. So HER2 positive clinically either 3 plus IHC or your FISH positive can either be your HER2 enriched subtype or your luminal B subtype. So not that all patients who are clinically positive for HER2 always fall into the HER2 enriched group. They can also be luminal B subtype patients. So this completes the discussion on HER2 enriched subtype. And the last and final is your basal-like subtypes, wherein there is very high expression of basal cytokeratin. That is how they are named as basal-like subtypes, and high expression of your proliferation genes. So these two are very highly expressed, but then they do not have your ER related genes and they do not have your HER2 related genes. And there is also parallelly loss of genes which are associated with cell cycle control, like your cyclin genes and the other genes which are associated with cell cycle control. There is loss of expression of such genes. The basal-like subtype or the triple negative subtype has further classification, which is again molecular based, as basal-like 1 and 2, the immunomodulatory, the mesenchymal and its mesenchymal stem cell-like, and the luminal androgen receptor. So there are 6 subtypes in the basal-like or your triple negative subset. So let's see the gene expression profiling of this. This is how it is. So this is the first one, that is the basal-like 1. This subgroup denotes basal-like 1. Here this denotes your basal-like 2. This is immunomodulatory. This is your mesenchymal stem cell-like, and your androgen or your luminal androgen receptor subtype. These are subtypes of your triple negative breast cancer or your basal-like subtypes of breast cancers. So TNBCs have all these 6 subtypes as of now, which is defined molecular wise. So TNBC means, what is it? It is a triple negative, that is ER, PR and HER2 negative. All 3 are negative for us. So most of them will be basal. At least 20 to 30% of even a TNBC, that is ER/PR HER2 negative, can still fall under your luminal molecular subtype. So not always which you see clinically, that means your pathological diagnosis of your either TNBC or your HER2 positive does not always correlate with your molecular subtypes. There can be interlinks and can be HER2 enriched can have a IHC which is negative, and IHC which is positive can still be a luminal B. That is the same way a TNBC can most of the time be basal, and a very little time it can also be a luminal ARB or a HER2 enriched subtype. This denotes only your gene expression or the gene expression profiling. So let's go. This is your triple negative, and most of them are basal-like. There are also few which can be either HER2 enriched or your luminal subtypes or your normal-like subtypes. Same way, when you take the basal-like, most of them are triple negatives, that is hormone and your HER2 negative. There can also be basal-likes which are hormone positive HER2 negative, which are all positives, or which are only HER2 positive. This is how there is interlink between your subtypes in the molecular level, though we can clinically classify them broadly as your hormone positives, HER2 positives, and your triple negative breast cancers. So let me recapitulate once again. So there are clinical classifications of breast cancer as your hormone positive, HER2 positive, and your triple negative. Also the other group is both your hormone and your HER2 positive. This is your clinical classification. This is based on your immunohistochemistry or your IHC. But your gene expression profiling has other 4 subtypes like your luminal A, luminal B, the HER2 enriched, and your basal-like subtypes. So these are the 4 molecular subtypes of breast cancer. Molecular is based on your gene expression profiling. Most of the times this clinical, the IHC or the FISH study correlates with your molecular subtypes, but there can always be no correlations between these 2 categories. So let's look at the same sentence again. So this is a triple negative and this is basal-like. So this includes both your triple negative and basal-like. This is the subset wherein there is triple negative but they are not basal, and this is basal which is not triple negative, and BRCA which is both basal and triple negative is this subset. So this is how there is interlinks between your molecular subtypes. Let me tell one word about what is BRCA-ness. We know what is BRCA, what is BRCA mutation, and what are the characteristics of a patient who is BRCA mutated. We have seen that they are usually young, they have high grade tumors, and they have basal-like protein expressions. A patient who is not BRCA mutated but who has phenotype of all these characteristics which is present in a BRCA mutated patient will be called as BRCA. So a patient who has a very high grade tumor, who are hormone and HER2 negative, who has pushing margins, very less of ductal carcinoma in situ, who has lymphocytic infiltration, has a basal phenotype along with other characteristics will be called as BRCA-ness or your BRCA-ness. This is helpful in predicting your sensitivity to the platinum and your PARP inhibitors. So, as we have seen, BRCA mutated patients are sensitive to platinums and your PARP inhibitors. So, let's classify this. This is the one which we have seen so far, the luminal, A and B, the HER2 enriched and the basal-like, the molecular subtypes of breast cancer. So, the ERPR expression is very strong in luminal A, variable in your luminal B. It is either positive or negative in your HER2 enriched and it's negative in your basal-like subtypes. The HER2 expression is very rare in your luminal A. It is very small percentage can be positive in your luminal B. It's very commonly expressed in your HER2 enriched and is again negative in your basal-like subtypes. The very high grade tumors present with either basal-like subtypes or your HER2 enriched subtypes and very rarely in your luminal A subtype. The P53 mutation again is common in your HER2 enriched and your basal-like subtypes. The high proliferation index, that is Ki67, is present at higher percentage in your HER2 enriched and your basal-like subtypes. Also, your DNA copy number, there is aneuploidy and genomic instability in all your luminal B, HER2 enriched and your basal-like subtypes, whereas your luminal A, which has a very good prognosis, is usually diploid. So, the gene expression profiling we have seen so far. So, let me recapitulate. What we have seen so far is the transcriptional or the gene expression profiling in your breast cancer. So, the gene expression profiling, we saw initially how it is being done, how you calculate or how you find out the gene expression of a tumor sample, mainly through your microarray. We saw the mechanism or the technique by which we do this microarray. Then we saw by taking a breast tumor sample, how it is being classified according to this gene expression profiling. We saw all the 4 subtypes or the molecular subtypes of this breast cancer. Now we will go on to find out what is the use of this molecular or the transcriptional profiling apart from your classification and prognostic purposes. Are they useful therapeutically? So let's go on to the other uses of this transcriptional profiling in breast cancer. That is, how do you utilize this gene expression profiling apart from finding out the prognosis of these patients? So we know that it was used for defining the molecular subtypes. It can also be used in determining the prognosis. And we should not forget that it is also useful in finding out or determining the value of systemic therapy, whether a patient will benefit from chemotherapy or endocrine therapy or both. So this is how your transcriptional profiling of tumors is helpful. So let's go, how are they useful therapeutically for us? So benefits for your therapy. The initial one which came up was your mammaprint, which we will see later. We will see in detail about the 21 gene assay or the 21 gene recurrence score. The 21 gene recurrence score is otherwise called as Oncotype DX. So gene signatures to determine the prognosis of patients with hormone sensitive tumors who are destined to receive anti-estrogen therapy. So a patient who is hormone positive and also HER2 negative, we will be treating with your hormonal therapy. Whether at all these patients require or not chemotherapy, we can find out from their gene expression profiling. That is the use of this 21 gene recurrence score or your Oncotype DX. It is done by your multiplex RT-PCRs and your paraffin blocks. This scoring was done initially with 16 cancer related genes and 5 reference genes. And the initial score through this retrospective studies was scored from 0 to 100, and the scores were less than 18, 18 to 30 and 31 to 100. So the recurrence scores were called as low when they were less than 18, they were called as intermediate when they were 18 to 30, and high scores are between 31 to 100. So this was from your retrospective studies. Then came your prospective studies, the TAILORx and your RxPonder, wherein they used the samples of patients and prospectively found whether they at all need chemotherapy or not. In the prospective study, that is the first TAILORx study, they used scores less than 10 as low, 11 to 25 as intermediate, and more than 26 as high risk. This is in contrast to the initial study, which was a retrospective study, wherein the cutoff were 18, 30 and more than 30. Here the cutoffs are less than 10, 11 to 25 and more than 26, more than or equal to 26. So this will be called as a low risk group, this as the intermediate risk group, and this will be called as the high risk group. So any patient who is particularly only node negative patients who are hormone positive and HER2 negative. So the patients who are taken are hormone positive breast cancers, who are HER2 negative breast cancers, and they should be node negative, that is N0 disease, up to T2, that is anyone who requires therapy and also who is not T3, which is not taken into this study. So anyone between T1C to T2, also with your high risk T1Bs. These are the patients who were studied in this TAILORx study. In this TAILORx study, the study was mainly for your intermediate subgroup. Those in the low subgroup always received endocrine therapy. Those in the high risk subgroup always received chemotherapy with their endocrine therapy. But those who are in the intermediate risk subgroup, the study was to find out if these patients at all need chemotherapy in addition to the endocrine therapy. So the randomization was between endocrine therapy alone versus endocrine with your chemotherapy. It was to find whether there is this endocrine is not inferior to this combination with chemotherapy. So it was a non-inferiority kind of a study to aim at your endpoint, that is your invasive disease-free survival. That is an endpoint which was used, and basically what they tried to prove was that your endocrine therapy is not inferior to an endocrine with your chemotherapy combination. So this was a positive study. It means that in this intermediate scores, that is between 11 to 25, only endocrine therapy is not inferior to endocrine with your chemotherapy. So it is a positive trial. It means that now in this subset of low, we can still do only endocrine therapy, and in this intermediate subset also we can do with endocrine therapy and do away with your chemotherapy. But then are we confident in not giving chemotherapy to all these patients? No. There is a subset analysis which was performed, and those patients who had a score between 16 to 25, the intermediate was 11 to 25, in this among that 16 to 25 group and who are less than 50 years of age, chemotherapy had a benefit in this subset of patients. So if you have a recurrence score of around 16 and your patient is around 45 years of age, we still do chemotherapy along with your endocrine therapy. So this is the subset analysis which shows that chemotherapy benefits in few subset of patients even in this intermediate risk subgroup. So this is how a TAILORx study answered the question of the requirement of chemotherapy in the intermediate risk subgroup. Now coming to the RxPonder, it is nothing but the same subset of patients who are node positive, that is 1 to 3 nodes who are positive. Again, when the same subset, that is between 0 to 25, here we took up the value of recurrence score from 0 to 25 and randomized them between your chemo or endocrine alone. And again, a postmenopausal patient, here only a postmenopausal patient had the benefit of only endocrine therapy in patients of score up to 25. It means the low risk subgroup and the intermediate risk subgroup in a postmenopausal women, either they are lymph node negative or positive, can undergo only endocrine therapy and they can do away with chemotherapy if their risk scores are either low or intermediate risk score. Obviously, for a high risk score, we will be giving chemotherapy. Either they are node positive or node negative, either they are premenopausal or postmenopausal, we will be giving chemotherapy in a high risk subgroup. The risk here is more than or equal to 26. So this subset all will receive chemo. But in the premenopausal patients, if they are lymph node positive, irrespective of the risk, that is even in low risk or in the high intermediate risk, we are supposed to do chemotherapy because they do have a significant benefit with chemotherapy along with endocrine therapy if they are premenopausal. So even if it is node positive or negative, if they are node positive, even your low risk had benefit with your chemotherapy. If they are node negative, the intermediate risk, the subset which we discussed now, had benefit with their chemotherapy. So this is how we know that a gene expression profiling is helpful in deciding on treatment for an individual patient. Whether or not to give chemotherapy, we can look at the score, we can find out what score and what risk they belong to, and whether they are premenopausal or postmenopausal, whether they are node or positive or not. Accordingly, we can find whether they at all need a chemotherapy. If they don't need a chemotherapy, we can confidently do only with endocrine therapy, and the chances of recurrence for such patients are very low. So there are other such recurrence score calculators like your 70 gene assay, which is through MammaPrint, which is through a study called MINDAC. Here the results showed that still endocrine therapy is useful in your low risk subgroup. The risk calculation and stratification varies for each and every assay which is being performed. The others are your PAM50, your 12 gene risk score, or your 2 gene risk score. All of these were performed in hormone positive, HER2 negative, and your node negative subset of patients. So this is how you utilize your gene expression profiling in your therapeutic purposes. So finally, to end with the case, so here is a 45-year-old woman who is premenopausal, who is T2 and node negative, who is hormone positive, HER2 negative, has a Ki67 of 8%. First, let's see what is this gene expression profiling helping us. We know that this picture per se will put your patient into luminal A. Here we have seen only the IHC part. With this, we are able to predict that their molecular subtype will be luminal A. So this patient, we can do your Oncotype DX, that is your 21 gene recurrence score. Find out the score of this patient, and having said, now let's take this patient is having a score of 17. Will we give or not chemotherapy? For sure, you are going to give endocrine therapy. There is no doubt in whether or not to offer endocrine therapy. That is a must. Whether or not to offer this chemotherapy becomes your doubt. We have seen in the subset analysis of your Taylor-X study that anyone between 16 to 25, as this number denotes, and anyone less than 50, even if they are in the intermediate risk subgroup, we can give chemotherapy, and there is benefit of chemotherapy. This is how the role of chemotherapy is being decided based on your recurrence scores or your gene expression profiling